Sure. So hello, everybody. Thanks, everyone, for being here today. It's a great pleasure to have as our guest today, Dr. Arthur Olson from the Scripps Research Institute. He's very well known in the molecular graphics, in the docking, in the modeling community. He has received his PhD from the University of California, Berkeley in 1975. And since 1981, he became a professor at the Scripps Research Institute. He has received several awards and has very interesting professional activities. I want to mention just a couple of them. He was the chair of the Gordon Conference on Visualization in Science and Education, which I thought is super interesting. We see from his work a lot of interest and contribution both in science and towards education and outreach, which is super important. And he has also been the director of the NIH Center for Structural Biology of AIDS since 2012. As I mentioned, Dr. Olson is very well known for the work with the Autodoc suite, which includes Autodoc 4, Autodoc FINA, and tools for visualization. And more recently, as he has just mentioned, they have been also developing programs, versions of Autodoc, which allow work with flexible receptors and with peptides, which I'm sure is of interest for many people here. And just to highlight the impact of Autodoc, the two papers most cited from Dr. Olson are the one that described Autodoc FINA with almost 18,000 citations, and the one describing Autodoc 4 with almost 14,000 citations. And that contributes for him to have over 70,000 citations and an H index of 70. More recently, his group has been working in novel computational methods for constructing mesoscale models of cellular environments, and that will be the focus of his talk today. So looking forward to hear about that. And with that, I want to say that it's a great honor to have you here, and I'll pass the word to Dr. Olson.
Okay, yeah, thank you, Rafaela. Yeah, it's really a pleasure to talk to you. It would be more of a pleasure if I were in Brazil, but you know, I think I'm reaching more people this way than I ever could have otherwise. At any rate, yeah, what I want to talk to you about today is what we've been trying to do in terms of looking to the future of structural biology at the molecular level. But first, I want to kind of step back a little bit to talk about a revolution that happened when I was, you know, basically a graduate student and postdoc, which was the switch in how, as a crystallographer, we built the models from the diffraction data. And what you see on the left is a picture of Bill Lipscomb, 1976 Nobel Prize winner, fitting a map, an electron density map that was computed from the diffraction data and then transferred by undergraduates with pens, marker pens, surrounding layers of density that was computed and printed out on a line printer. And those were stacked in 3 dimensions. And what you saw was a 3-dimensional electron density map, like a topographic map of sections going through space. And what you did was look at that through a half-silvered mirror, and with the appropriate lighting, you saw the map, which was stacked inverse, mirror image, and you built a physical model out of brass parts, perceptually putting the parts into the electron density. And that's the way all the structures were determined. But in fact, building the model wasn't the only problem. You had to be able to then measure the atomic positions, and we did it with a sight line and a plumb bob projected to get the coordinates of each of the atoms. So at that time, 3D computer graphics was just being commercialized. And it turns out that crystallographers were among the first people to realize that you could put an electron density map up in 3 dimensions and fit electron density maps on the computer screen. And what that did was 2 things. First of all, it represented a killer app for the incipient 3D graphics commercial enterprise. So in fact, we were the only people that, as scientists, that had the budgets to pay $200,000 or $300,000 for a graphics system. This is without the computer behind it. You had to buy that separately. In today's market, that would be $2 to $3 million US dollars. So they needed some markets that could actually allow them to develop the technology. And it turns out that protein crystallography was a major market for driving the industry. But on the other side, it changed the rapidity and ease of actually determining the 3D structures. So the technology, the computer technology, was both a killer app for the technology, and for the science, it was killer tech for the science. It really changed it. And you could see that in terms of the numbers of protein structures that were being solved and the rate they were being solved before and after the advent of electron density fitting. So the one thing I could say is that we're in a situation that's quite similar. History might not repeat itself, but I think it, as the saying goes, it rhymes. And that is that there's technology, computational technology and visualization technology that is having a sincere impact on the way science is done. I mean, as bioinformatics, you couldn't do your work without computing. And the size of the data and the variation of the data is exploding still. So the way I look at it is that we're now kind of living in an age where we can start to appreciate life and living systems from a molecular viewpoint in terms of the structures and the dynamics of those structures at the molecular and atomic level. If you look at the systems that we're talking about, well, certainly they're governed by all the laws of physics. There's, but in terms of complexity, you also have to look at it in terms of the chemistry because of the combinatorics. Physics talks about elementary particles and so forth. Chemistry talks about the combinatorics of atoms, which is enormous. And so that adds a higher level of complexity to the systems. And likewise, biology, because the chemistry that we're talking about is influenced by the history of the molecules themselves and their environments, then the complexity gets even more enormous, as we're all aware. But in fact, the belief that there's a physical continuity from atom to organism means that we should, in principle, be able to model the mechanisms of life. And by modeling, you have to talk about the kinds of modeling that you need. In physics, typically the models are analytical models. You can write them down as equations and simple variables relatively. Chemistry, you have to include the mass action or the thermodynamics of large numbers of interacting particles. And in evolution, you have to use multi-scale and multi-mode models. No single model is going to answer all of your questions. So the way I frame it, as far as the challenges that we see in trying to understand the complexity of the molecular cell, has to do with how that we interact with the data, how we develop and utilize and interact with our software, how we simulate the dynamics of these systems, and also extremely important, how does the human interact with and understand things that are this complex? So for data interaction, I think you have to think about integration. How is it that you can integrate information from different scales? Since evolution itself is scale free, there isn't a single view that can tell the whole story. In terms of software interaction, reuse and collaboration is a must, as you see. I mean, we're all dependent now upon layers of software that have to interact for us to do, simple things like look up Google on your iPhone. So what we can reuse and how we bridge different disciplines rather than reinvent everything is an important part of the technology. In terms of simulation, obviously you have to think over multiple timescales and understand that the phenomena that you're looking at is not an equilibrium situation. It's a flow of energy through the mass that the physical mass that is represented. And then finally, in terms of human interaction, I think we need to think about more than just what our eyes are seeing, but more what we're perceiving through any and all of our senses. And so I think that's an important part of understanding and communicating complex systems. So first of all, what kind of data interaction are we talking about? Well, you can think of what's called integrative modeling as a form of data reduction, where you have data coming from lots of different sources, some of them structural informational sources, some of them spectroscopic and other types of experimental data can be embodied in a structural context. And the structural context tells us lots of things, and also allows us to integrate in that structural context, or inquire into that structural context. So basically, we coalesce all the known information about a system into an easily interpretable model. And the information is used not in isolation, but simultaneously to generate the model. So the kinds of data, so first of all, you can look at the problem of living systems from two ends, from the atoms to cells by building up living structures, or from cells down to atoms by looking at them in context. So there's lots of sources of both structural data, X-ray crystallography and NMR spectroscopy. And now one of the revolutions in the last 10 years is certainly cryo-electron microscopy, which is now in essence taking over from X-ray crystallography in an area of scale that is, instead of single protein level, it's multiple or complex proteins, multiple protein complexes. And there are other tomographic methods, again, that can give us information at the atomic level in terms of by cross-linking or affinity purification, or fluorescence methods like FRET, site-directed mutagenesis, and so forth. And also the integrative methods or the computational methods can all be based on atomic level information, like the protein structure data bank and methods like computational docking and so forth. But the other way you can look at it is from the cells down to the atoms. So there's enormous amounts of data being generated at the cellular level, from cryo-electron microscopy and X-ray tomography, along with the amazing advances in fluorescent imaging and methods that can tell us about the genome architecture like Hi-C and so forth, and some computational methods like I'm talking about, integrative structural biology and proteomics and computational systems biology and so forth. So these are all views from the top down, and they tend to be in context. In other words, they represent the cell and its contents and looking at it in situ. From the biological realm, also, obviously, things that that we call life happen at all time scales as well as length scales. And so when we're trying to model the environment, it depends upon the scale in which we're looking, what kind of models we can use, going from the very finest models, the quantum models, to more force field based mechanical models, and then up through what I would call, a range of coarse grained models, and then in the macro scale, more continuous methods, or let's say finite element analysis type of models. So all of these models can span time scales from femtoseconds to years. But if you're looking at a phenomenon, you need to essentially inform each model from the one that's higher. It's kind of what's called a handshake region. So you go from, electronic structure up to rigid multibodies, et cetera. So I think it's important to recognize that the real issue in any of these kinds of models is how they depend upon and how, for the particular phenomenon you're looking at, how well informed is it of the scale, the time scale above and the time scale and below. So just as an example, how do things happen at different time scales? Well, here is a kinesin walking on a microtubule. And as you see, I play it again, is random motion, but there's a flow of energy using ATP and converting it to ADP, which changes the conformation of the kinesin and allows it to move along the microtubule. So it's not like it's just walking. There's constrained random motion that's where an energy flow changes a conformation and allows a binding event to take place. It's also, you should notice that the microtubule has a directionality to it. So it's not, there's a beginning and an end. And so there's a directionality to the motion itself. And so the reason I mention that is that this is an example which caught my eye several years ago of an extremely simple system made up of 4 different kinds of molecules that where you can see the relationship between the dynamics at different scales, and you can model it. So what are the components? Well, the components are the tubulin, which make these microtubule fibers. And then there's the kinesin, which is the motor that walks along in a particular direction. And then there's what's called a biotin-streptavidin system, which enables multiple kinesins to link together. So they can span between adjacent microtubules. And each of these microtubules has a directionality. So these things can either walk in parallel, whatever tubes come together can either walk in parallel or they can walk in anti-parallel. If they walk in anti-parallel directions, then that tubulin extends and becomes longer. And then it gets so long that it reaches its buckling limit and it buckles back on itself. And then it starts to tread again out to that buckling limit. So what happens if you, and then the only other ingredient that you put in is a crowding agent like polyethylene glycol, PEG, that allows these tubulins to kind of associate or pack together into fibers. So what happens if you put these things together? If you see on the left, well, nothing. But if you add ATP, depending on what the concentration is, you get this kind of motion, which is called the dynamic pneumatic phase. And it's basically a motion that's due to the extension and buckling of these microtubules. If you put it into a droplet of water, it's let's say it's an emulsion of water in an oil context, you get this kind of motion on the surface. If the drop is big enough for the thing to buckle, if it's too small, then it frustrates the dynamics and it doesn't go into this treading mode. So what you get, is this kind of motion of these cell-sized droplets. If they're too small, they don't move because the thing doesn't move. So you get macro scale phenomena from what you know is a very, very simple molecular system. If you put the system into a membrane, you even get these pseudopods that stick out and looks even more lifelike. This is just a very, very simple system, but people might think it was a cell. So like I say, we see things at the atomic level, and we can see things at the cellular level. So this is the nanoscale and the microscale, but there are levels in between where you're looking at things like viruses and so forth, where you either have to isolate parts of them and look at them independently, or you have to basically from this side look at it in a very fuzzy way. So here you have sparse models, and you also have fuzzy observables because of the limitations of either the optics or the isolation methods to get the individual components. So this is an area that is called the mesoscale, and what it means is that you have to do data integration to get a picture of what is actually happening at the molecular level, because you can't get it directly from any single experiment. As I was saying before, you integrate all of these various sources, like which memories which proteins are in the system, where they are and so forth, how they're distributed, what the shapes are. And then you put this together into a picture or a model that is consistent with all of the data. And that's something that David Goodsell pioneered when he was a postdoc in the lab. The picture on the upper left is one of his first drawings, kind of asking the question, well, how crowded is the molecular environment? What does it look like? And given what we knew in 1991, this was kind of the resolution of the components that he was aware of. And it's interesting to look at his paintings subsequently. This one is from 1999, and this one is from last year. And if you look at it closely, you see how much more information is known about each one of the components in terms of where they are, what their numbers are, what types they are, how they're shaped, and so forth. So this indicates how much information goes into these representations. And in fact, the work of making one of these paintings is not in the painting, even though the painting itself does take significant amount of time and an amazing talent in terms of coloration and so forth. That's the artistic part. The real hard work is actually in getting the data together and knowing what these structures look like at various scales and how many of each there are, et cetera, et cetera. In fact, I want to make some points because I think that David's work has been highly influential in terms of how people are starting to view large molecular systems or molecular systems in the context of living things. This is a series of paintings that David did for our center our HIV center, and the drawings are available there. But the point I want to make is that each one of these embodies the current state of knowledge of various processes of HIV as far as people knew it or know it at the time. These are frozen in time in terms of the states of knowledge, but they embody the current, then, let's say 2 years ago, the current thinking of what budding looks like at the molecular level when the virus buds out of a cell. What are the processes that take place in terms of the maturation of the virus, going from this immature state to a post proteolytic state that reforms into the mature virus that's ready to reinfect. And then how the genetic information travels in the host cell, and the reverse transcription that takes place inside of the capsid core particle to convert the RNA genome into DNA, and then how that gets integrated by going through this the nuclear pore and basically sewing itself in, integrating it into the host DNA, and then the RNA transcription that takes place from that piece of DNA, and then the expression of the proteins for the budding to take place again. So again, these pictures embody a wealth of knowledge. In fact, when you showed it to one of our face-to-face meetings, it got one of these got more discussion than anything else, because like in science, not all the rules are in and not all the facts are in. And so there are disputes about certain things, but these pictures were able to visualize a particular thing and really catalyzed a lot of discussion. So I was also inspired by David's work, and we collaborated on a cover for a scientific report, a Scripps scientific report. I took one of David's paintings and took a slice out 75 nanometer cube, and used the concentrations to build a model, and then made a 3D lenticular print that went on the cover of the report. But the point is, it took me many weeks to put this thing together in terms of just getting the structures, knowing how many, and then actually the physical placement at high density was not trivial to do by hand, especially given the modeling tools that were available at the time. So it got us thinking about 3D modeling of cellular environments based on what David was able to put together. And part of it essentially was a packing problem. And so what do you need? What kinds of things do you need in order to build a physical model that looks like one of David's paintings, but is actually in 3 dimensions? And packing itself is at least packing of spheres has been a kind of a mathematical area of interest for years, and as are the kind of more procedural types of structure like L systems for trees and leaves and so forth. So there's been a lot of work on things like that, but putting them all together into a specific domain like molecular biology has not really happened in the same sense. So you need this volumetric packing and surface packing and procedural packing and generation of fibrous ingredients and so forth. So actually, so Graham Johnson came to the lab, again, inspired by David's paintings. Graham had worked as a scientific illustrator for 10 years and has a well-known cell biology book, the Pollard Earnshaw and book he did all the illustrations for. But he wanted to come and actually get a PhD in molecular biology. And so he wanted to actually develop this three-dimensional packing program. And so when he came, that was the beginning of the AutoPack and CellPack effort. So Graham basically, developed a method that defined a cube or a volume of space. And in that volume of space, there are different compartments. These are compartments typically that have membranes around them. There are also complexes that are large enough to actually see in tomographic representations. So you may have particular placements or numbers of large molecular complexes, but then you divide up the space into tiny cells. Basically, you put a grid on it, and each point knows its distance to the nearest object, surface. It's basically a distance field. And so you can use that and just start putting stuff in to the available space. If you start with the larger things first, then you can pack things more tightly and so forth. Fairly simple. And what you end up with is methods to pack things at very high densities. At the top, it doesn't make any difference what the shapes are, basically, as long as they occupy space that hasn't been occupied before. So you can have different sizes and shapes and so forth. This was called AutoPack. But then when you put biology into it by talking about having material or objects that only can go onto membranes, or you have long fibers or procedural fibers like chromatin fibers and so forth, then you need to have special recipes for that. And those recipes are basically what's called cell pack. You also obviously can determine gradients of different ingredients and so forth. So what you end up with is a tool that actually can take a structure like a HIV, mature HIV, which has a membrane exterior and then a capsid inner compartment. And then, so each of the points of where they are, whether they're on a surface or whether they're inside or they're outside. You have the list of ingredients and where they belong. You put it all together and you get a model that's consistent with all the data that you have. Now, each model that you make can be distinct from any other model because you're using random numbers to place things that are in well-stirred or areas where things are essentially randomly distributed. And what you end up with is a model that you can look at all the way down to atomic level, and it's made up of a certain number, a known number of each ingredient in the particular model. So here we're just adding it. This isn't the way it packs. It doesn't pack by ingredient, it packs by size. Okay, so we were interested in the immature Virion model, and there was a little background. There's a polyprotein called GAG, which basically forms together. It binds the RNA, preferably of the virus, and it forms this kind of bulge that ultimately pops out of the cell. The GAG itself forms hexamers. The the N-terminal domain called matrix attaches to the inner membrane, and it forms trimers. The next domain, which becomes another protein, is called capsid, and it forms dimers between hexamers, and the nucleocapsid binds the RNA. So basically, if you want to build a model, you have to find the structures of all of these things and put them into not only the right order, but in the right interaction network. And to do that, you can use cryo-EM and crystal structures to build the models into the electron density. And you can see the various interactions. For instance, this tight helix bundle at the P1 site, which actually is important in the maturation process. It's the last cleavage site when the protease is freed to convert this polyprotein into the individual proteins that then go on to make the mature virus. But the immature virus, there's some, this is a schematic based on a tomogram, EM tomogram of the immature virus. So these hexamers each represent a hexamer of these GAGs and also GAG-Pol proteins. And so Ludovic in the lab, so here you want another procedure, like a tree growing procedure. He developed a procedure where when you place a hexagon, you try to tile it and place it another hexagon next to it. You pick them randomly, placing hexagons, but on a sphere or an enclosed surface, you cannot tile that surface with hexagons. So what happens is that you end up with spaces. So this is the algorithm. You just kind of randomly place the next hexagon, and then it grows up to the known number of GAG molecules that are in the structure, and you end up with a structure that covers about 2/3 of the surface, but it doesn't cover the whole surface, and it has these breaks in them because of the packing problem itself. And in fact, once you have that, then you can put the atomic model into those hexamers, and then you have a model of an immature virion. So how do you use a model like this in terms of data interpretation, for instance? Well, so there were two things. First of all, there were the observations on the immature particle itself. And this was, as I had mentioned, from John Briggs. And what you can see here is that the hexagons pack in basically the same way that Ludovic Authin's model shows. In other words, there were the same kinds of breaks and so forth. Now, there was a spectroscopist who was using, or a microscopist using super resolution fluorescence microscopy called STED, which has about a 40 nanometer resolution. And he was asking the question, in the immature virus, well, back up a second, a lot of viruses have these spikes, like the coronavirus, et cetera. HIV has a spike on it that's called the envelope protein. And, but so it's what the immune system tends to see because it sticks out. But unlike the coronaviruses, HIV has very few envelope proteins on its surface, on the order of 10 or so trimers, as opposed to the 90 to 100 trimers on the coronavirus. So he was asking the question, how do these things assemble on the surface. How are the envelope proteins, the spikes, distributed on the surface? So he attached a fluorescent label to the spike and used STED microscopy to look at the accumulation of where the envelopes are. You couldn't see at 40 micron resolution to something that is only 40 nanometer resolution to something that is about 120 or 130 nanometers in diameter. All you can see are spots. And so he had 3 graduate students look at 100 different proteins using STED microscopy, and they counted how many spots they saw, whether there was 1, whether 2, or there were more than 2. And from that, they developed a model that they concluded that the envelope proteins were randomly distributed over the surface. We decided that we'd look at that problem from a modeling point of view. And so what we did was we generated, let me go back. Yeah, so we generated 6 different models, hypotheses of models, one of which the envelope proteins were distributed randomly, one where they were distributed in the open space in the surface where the gag didn't accumulate, one where they associated it in the gag surface, and one where they associated adjacent to the gag surface. In other words, they were they close packed against gag, and so they would be on the periphery. And then a couple of other hypotheses that had to do with how the envelope itself attracted itself, whether it was both an envelope attraction and kind of a close packing with matrix, or whether it was all close packing of the envelope. So those were the models. In each one of these models, we actually made 1,000 models, because we could make, and each one is randomly generated. So they're all distinct, but again, they have the same structural characteristics. So, and then you could take that and kind of make a synthetic STED microscopy by reducing the resolution of the models themselves. And so what you end up with are spots that look just like the experimental spots with 1, 2, or more than 2. And then you can analyze those things. Now, the difference here is that we actually made a 3-dimensional model. They were looking at projections. So we had X, Y, and Z views of each one of these. So we could analyze the number of spots in a different way by looking at them from 3 orthogonal views. And ultimately, what we came up with was a distribution of 1, 2, or more than 2 spots based on the different models that we had calculated. And what we found was that from what their observation was matched more, was not the random distribution that we found, but it was in fact a much different distribution than the random one of these, or because there's some ambiguity there. So modeling can help interpret information in a way that can go beyond other current methods. Another, this is just an ongoing thing, but I just wanted to mention it in terms of HIV work, is modeling how reverse transcription and may influence the disruption or the of the capsid. In other words, the capsid is this container that is released from the virus into the cytoplasm, travels to the nucleus, and during that travel, and now I think pretty much certain inside the nucleus, the reverse transcription has taken place in this vessel. And the question is, now the DNA has to be able to get out, so it has to uncloak the capsid. So what is the force? What happens at that point? How does it break apart? So we're doing a multi-scale modeling of this particular process. Christina Garza is taking the atomic level structure and doing steered molecular dynamics, pulling apart one hexamer from another hexamer to find out the force versus separation, where the breaking point is. And at the other end, Andrew Jewett is doing a coarse grain dynamics of model that takes a look at the difference in the inside and the outside in terms of large molecule mass and how it's organized, and what kind of results one might expect. And that's ongoing work, and so I won't. The results are still kind of being interpreted. Let's put it that way. So the next challenge, obviously, was to go from a virus to an actual autonomous living organism. And so we're interested, obviously, in doing the simplest thing. And again, this is one of David's paintings of a mycoplasma. And so that was a target that we thought was reasonable. First of all, it's small, and it is the simplest living organism as far as we know, because this organism has been engineered, at least the one that you're looking at here, has been engineered to have the fewest number of gene products. So it's the simplest one, and it's the one that Craig Ventnor and his team at Ventnor Institute have been working on as the first synthetic organism. There's cryo-ET images, and there's a whole cell model of a related virus. And this is a model that came out of Marcus Covert's lab at Stanford. And he contacted us because he had made a kinetic model of a mycoplasma gentilium. And so at each point in time, he could calculate the shape, the metabolic rates, the gene expression rates, the DNA replication. You can see the point at which one DNA is totally read and then creates the second one, and any of the translation, which proteins are actually produced at any given time point in the life cycle. So he contacted us and said, asked if we'd be interested in trying to build a model where you could look at the state of this virus at any time point. And there, you have first the challenge of knowledge. You have to get all of this data together in terms of what membranes are where and what structure can we use and so forth. And Ludovic Tan. So this is work initiated mostly by Martina Maritan, developed a program. Actually, it's a browser tool that anybody can use. It's called Mesoscope, and it allows you to import ingredients, let's say in a table, and with various characteristics like where they are, what size they are, what function they are, and so forth. It connects to the protein database, and connects to other databases that have all the functionality and so forth. At any rate, this has been used by Martina as a curation tool for each one of the proteins that are in the mycoplasma. So this is kind of a picture of where the structures have come from. So let's see, I just need to move this out of the way for me. So most of the structures this is the confidence. Unfortunately, it doesn't have the numbers here, but only, I think, 6% of the structures were actual structures from this organism that had been solved by crystallography. The rest of them are homology structures and or in-house generated homology structures. And this is just this is the palette of molecules that are in the mycoplasma. So, so as it says, there are only 9 experimentally determined structures, 494 homology models, and 147 homologs. And the homologs are basically the multi-protein complexes that exist in the database, but are not from that organism. So you have each one you have to decide on what is the best model and how much confidence you put in that model. The other aspect of the mycoplasma is that it has a much larger genome than certainly the small RNA genome of HIV, and the structure is much more complex in several ways. There are a number, obviously, of protein binding proteins, DNA and RNA binding proteins that you have to account for. And then there's the structure of the fiber itself. And it turns out that these bacterial structures and other structures form supercoils called plectinemes. And you can kind of map where these plectinemes are by looking at the RNA polymerase binding sites through a variety of sources. And so for the E. coli, this red line that goes on the outside, the areas that are red are non-plectinemic areas. They're not supercoiled, and therefore that's where the RNA polymerases are. And the ones that are supercoiled, where the binding of RNA polymerase is pretty much excluded. So you can get a map of where these supercoils are and how long they are, since you know the length of the entire thing. And you can start to actually make a model. And Adrian Alcock and his group were the first to actually make a model that started with the plectinemic map of E. coli, and then through a series of coarse grainings and minimum energy simulations and minimizations, was able to bring it down to the atomic level in his model. So all of that can be done, but it's fairly complicated. So when we started to look at the genome of the mycoplasma, it's got about 1 million base pairs. And so David came up with a quite efficient algorithm that that was used on the mycoplasma. The topology was known, it was closed circular. You could get the superhelicity or the plectonemic density through experimental evidence. You know what the persistence length is and the available volume, and the ChIP-seq and Hi-C data was available to get the placement of particular proteins and so forth. And the way it worked, is actually fairly simple. He developed a lattice, basically a rectangular lattice, and continually subdivided it. So you have through random moves along a 3D lattice, and you can grow if you know where, at any point in this structure, which is labeled with the sequence itself, you can add these plectonemes just as points along this lattice, and then you can extend that into a higher level representation where this line becomes a superhelix of a certain twist, and then you can subdivide that more into beads and then relax it. So what you actually can get is this type of representation of where the DNA is and how it's supercoiled. And this is kind of a 5' to 3' coloring here in terms of how things are distributed. But in fact, you know, for any model, you know where every gene product is. And so this is the lattice-based, one of the lattice-based models for the mycoplasma. And so you start out on a grid, and then basically you relax it. Each one of these is a known DNA or RNA binding protein. So they are placed in terms of where they are in terms of the expression maps that come from the covert model. So to do something like this, you set up a pipeline, and there are two routes to that. One of them is to generate the lattice nucleoids, and the other is to generate all of the other soluble and membrane proteins using Mesoscope. And like I say, each one of these can be looked at. Membrane proteins can be evaluated for whether they're oriented and in the right place in the membrane and so forth. And what you do is you integrate those two models. You generate a cell pack model. That generated model then gets refined in terms of overlaps and relaxed. And then you can export the model even into Molstar, which runs in the PDB as a browser. These are complex models, but you can load these models in. And you can analyze them in lots of different ways. So here at 3 different time points, frame 149, 1184, and 6973. So each one of those frames from the simulation, you can build a model, or you can build hundreds of models if you want. And each one you could look at, for instance, what percentage of the chromosome has been explored by the RNA polymerase over time, what kind of gene expression is taking place, again, seeing it with a color code or something, and also protein expression. So we're only just starting, we just published this paper at the end of last year. And I think that, this type of thing, this is a little movie that Martina made just to show the model, one model as it was built. So you see the membrane proteins. Now you're looking at the packing of the soluble proteins and the DNA and the RNA. I think it just kind of focuses in on a ribosome translating RNA, the message RNA. So these models, I think, are going to be very valuable in a number of areas, including simulation, image interpretation, and in fact, exploration and communication. So, which brings me to the last topic, which is human interaction. So if we think about cells, the size and complexity is enormous. So far, I've talked about HIV, which is a little dot that you probably can't even see. The mycoplasma is at this scale just a dot, compared to one of the smaller human cells, a pancreatic beta cell. So the number of atoms goes from 10 to the 9th up to 10 to the 14th. The amount of data goes up by a factor of 1 million. Protein types is up to 10,000 as opposed to the 500 in mycoplasma. And the number of compartments goes from 1 or 2 to thousands. So how do you deal with this stuff? This kind of complexity in representation and navigation and so forth. So first of all, you ask the questions: what, when, and why. What level of detail do you need at any particular viewpoint, at any particular time of an interaction with such a complex model? When should you actually create the data that you're looking at, let's say? So it's certainly possible in these environments not to create an ingredient until it is in a frame where you need to see it or inquire it. And why? The other question is, what's the question that you're asking? And the representation can certainly be changed to meet those needs as opposed to meeting all possible needs. So this is where technology has come in and what's happening in the computing world since 40 years ago. First of all, graphics processing units and these special purpose processors that can, for certain tasks, do a lot of parallel processing, basically have taken off. These companies like NVIDIA have become dominant in the computer chip business because of the GPUs that they have developed. And certainly it's been driven by games and movies in terms of the ability to interact in real time with large amounts of data. So the GPU is important. And from a software development point of view, as I mentioned early, you don't have to reinvent everything. And in fact, there's software development environments now that have enormous types of resources and structures to access lots of data and assets, as they call them. So game engines can greatly facilitate their software development and content creation, and all the stuff that we've been developing in terms of CellPack has been using a game engine called Unity. These game engines have built-in constraint modeling and physics engines. They're optimized for GPUs. They're cross-platform compatible, and they're targeting consumer-grade technology, which means there's a huge development and maintenance effort that goes into it with very large commercial backing. I mean, the game industry is larger than the movie industry now, as far as the United States is concerned. So what are some of the tricks that are used for looking at huge amounts of information? Well, one of them is what's called level of detail. You only need to have detail on things that need the detail. Something that's close to you should be represented maybe at the all atom level, but then you can go down to lower and lower levels of detail the further away you are. An object that is smaller than a pixel on a screen or some kind of display is, you can't, that's undetectable. So this level of detail is one thing that you can do. The other thing is how, for instance, if you have a bunch of spheres as atoms, how do you represent each sphere? Well, geometrically, you would have to have some kind of a triangulated surface and so forth for a sphere. But in fact, from any given viewpoint, you could render an imposter that is actually just a texture map on a single polygon that looks like a sphere. So as far as the visualization is concerned, you can greatly reduce the overhead by using imposters. And so in collaboration with a group from Ivan Viola's at the Technical University of Vienna, they were developing CellView with GPU performance of 10 billion atoms at 60 hertz. And that was implemented in Unity. And they contacted us because they were interested in using some relevant biological systems and of high density. And so we started collaborating with them. And this is just a kind of a stress test, I think, with 10 billion atoms in this cube. So we also got, in collaboration with Ivan's group, got interested in re-implementing CellPack on the GPU. And Ludovic Autain in our lab came up with a scheme where you had rapid volumetric definition in arbitrary compartments and instant placement of each of the ingredients, the protein ingredients, by random access to what's called a Halton sequence. And I'll explain that in a moment. And also, you would generate fibers independently and integrate them as we had done, as we did with the mycoplasma model. Also, the instant packing involves a kind of a different level of detail, which is the level of detail of collisions, how you represent the shape of a complex molecule with various geometric representations that can be computed very quickly on a GPU. And so we have lots of ways of making spherical clusters at various resolutions, depending on, again, on where you are and what you need. And so from this, and also the question of how you generate lipid membranes. And that uses something called akin to lipid wrapper, where you pre-compute a large raft of lipid and you minimize the interactions. And then when you have a surface you triangulate the surface and use each of the triangles as a cookie cutter to cut out a group of lipids on that surface, and then you tile it that way. And that works fairly well. There are some issues around it that are still being worked out. But what you end up getting is what we call instant packing. If you have the number of proteins and you have the distributions and where they are, and basically by changing a random number, you're changing the placement of each one. So this is kind of real-time generation of different models of the same protein state, or the same virus state in this case. Let me go forward here. So in Ivan's group, Tobias Klein took up the challenge of using the GPU to generate fibers such as DNA and RNA. And what you see here is, again, real-time interaction. It's hard to see these sliders over here, but each one of those is changing a different aspect of a model. So in fact, you just went from a small piece of the DNA to the large one. You can also place these plectinemes anywhere along the sequence, and they can be as long and as meandering as you want. And you can even change the helicity, basically the looseness of the plectineme and the twist. All of it is kind of real time. So so this is an interesting development to speed up the whole process of generating these models. As you can see, you can place the plectinemes anywhere in the sequence, and this is done at the atomic or the sequence level. So the representation can be beads, but it can also be the full DNA representation. And so you can see here, by moving these plectinemes along, you're changing the model at the DNA level. So with these representations, you can envision generating these things very quickly. Let's see. Oh yeah. So here he's just this is just a part of what's been generated. So if he goes and he extends it all the way out and puts the atomic description, that's the final. This, for instance, is E. coli, which took Adrian Alcock, I don't know how long to generate. And so our attempt to go to human cells is a modest one in one sense, but I think also an important one. And that is that we started to model the insulin secretory granule, which is, this is data that comes from soft X-ray tomography. This was done at Berkeley Labs with Carolyn Larabelle. So it's a relatively low resolution method, but in fact, we can use that to the segmented volume to define a volume that we can use at the cell pack level. So we can go down to the individual insulin crystals and granules and show it in the context of the beta cell from data from much lower resolution. And in fact, let's see, how that stopped. I'm sorry, I lost my. Sorry, it's frozen somehow. gonna have to get out of. Am I still there? Hello?
Hello.
Yeah.
We can see the slide, but it's frozen.
Yeah, I can't, I don't know what happened to my computer. I can't even escape. Can you stop my share for a second, just so I can, oh, wait a second. Did it change?
Yes.
Okay, good.
It did.
I don't know what happened.
Perfect.
Okay, I'm good now. Thanks. So at any rate, just to mention that how we can use this kind of data and how we're using it. One of them is, again, to generate the soft X-ray tomograms using the models that we have. So we can change the size and the shape and so forth, and then simulate the tomogram and use that to do the interpretation and the reinterpretation of tomograms. And so it's actually turned out to be fairly useful because these insulin secretory granules mature from no crystal to a large crystal of insulin. And what we found in the simulation was that they're probably in the X-ray tomogram not seeing a lot of the immature granules because most of the contrast is due to the crystals. At any rate, the last thing I just want to mention is kind of more outreach and user-friendly tools. A lot of people admire David's work, and so recently he, along with Adam and Ludovic, developed a program called Cell Paint, where the palette is the molecules, and you can draw membranes and you can populate areas and membranes and so forth. So the image on the right was done with Cell Paint in probably, maybe a half an hour compared to the amount of time that David takes to paint one of these. And this is available both as a browser tool at cellpaint.scripps.edu. So that's gotten a lot of attention lately. It was just published last year. But it uses, the nice thing is that you can make diagrams with it, and you know that the scales of all the ingredients are correct and the shapes are correct and so forth. And it uses the tricks that David uses in his paintings. And finally, one thing that I've been working on with Ludo and Adam is a VR version of Cell Paint. So here, what you're doing is creating a three-dimensional representation where you're painting with molecules. This happens to be spectrin network in a red blood cell that I created probably in about a half an hour with the ingredients that are involved in the spectrin interactions in a red blood cell. And you can see that this is just capturing, obviously, a small part of the immersive experience, but each one of the objects can be seen down to atomic level, and they could be manipulated and drawn. This was already created. I'm not showing any creation here, but just modification of placement. But one can generate three-dimensional landscapes and use them basically either as the beginning part of a simulation or as a way to communicate certain processes like David's drawings. So thing is that there's full body involvement as far as the perception there's definitely a heightened sense of three-dimensional and spatial relationships, and the interactions are very natural. Nice thing about this is that these are commodity-based systems. I mean, you can buy this system, the VR part of the system, for $300 on Amazon. And the tool itself allows interactive environment building and membrane construction and fiber drawing, multi-scale viewpoints placement, and also has Brownian motion and that you can put into the environment. So with that, if I can just thank everyone that's in the lab now, my three co-PIs, David, Stefano, and Michelle, and postdocs and graduate students that did the work that I talked about today, and also the group of Ivan Viola at the Technical University of Vienna. So I know I've gone long, but I'm happy to take questions if there's time.
Hello, thank you so much. This was an amazing talk, really interesting. I have to say, it's a bit overwhelming to think about generating this model and all of the challenges to integrate the data. And I thought it was also fantastic to see how you combine the scientific accuracy with the perspectives of a artistic representation that really makes people interested in exploring this model. So it was really very interesting.
Thank you.
So congratulations, thank you so much. And I would like, let's see if we have questions. I'm looking here. So while we wait for further questions, I was wondering about how when you're building these models these days, you have shown a lot of development, several software you have already worked on, and you even mentioned a pipeline at some point in your talk. So I'm thinking, how automated is the process now if you want to build a model for a given virus? How does it work? Is the challenge mostly in getting the data, or once you have the data, do you still have to work on a cases by cases to improve your software and build the models?
So the short answer is it's not automated as much as we would like it to be automated. But in fact, you're absolutely right, but the work is in getting the data that you want to use. In other words, once you have the data, so for instance, as an example, with the mycoplasma work, okay, so we've got the data not just at one time point, but at all of these time points. So that data, once you have the data for one time point, in other words, you have a recipe, you have a set of ingredients for the model, which is the proteins and the fibers and so forth. So creating the second time point or the 10th time point is easy or relatively easy. Once that data is actually there. So here the data that we have is generated by a model. So once you have the proteomics and you have all of that information, the model just tells you what's being expressed when and so forth. But you know, Martina worked for 2 years to curate the mycoplasma model. One thing that I will say, though, is that with the advent, which I think is an amazing revolution of the AlphaFold approach, a deep learning. So the revolution that I was talking about that's taking place now is both a revolution in experimental methods like electron tomography and microscopy, but also in computational methods like these deep learning methods. And the two go hand in hand. The reason that AlphaFold works is because there are 200,000 structures in the database. And so the ability, so if Martina were doing this work today, as opposed to 2 years ago, the AlphaFold would probably have cut her work maybe in half. I mean, nothing is perfect. You still have to do the curation in some sense. I guess the saving grace is that we know, well, even though there are lots of proteins, even humans only have about 20,000 different protein types in their bodies. So there is a limit. And so I'm fairly optimistic that these things will become more automated and faster, just like the cell pack has become very fast. The problem, though, is, and this is really an informatics problem, a fundamental informatics problem, is how is it that you can integrate all of this data into one place? In other words, we're in a state where there's a bunch of different states, and each one is generating their own resources, but we're not. It's not easy to put it all together right yet. So that's an area that needs a lot of thought, a lot of work.
Yeah, I imagine. So we have several people here congratulating you, saying that your talk was really nice. And we also have some questions from Gregory Keaton. With a cytoplasm so packed, how can signaling efficiently occur between the cell surface and the interior of the nucleus? Is there some mechanism we are missing? Do your models predict anything?
I can answer that generally, which is yes, there are lots of mechanisms that are missing. Now, I'm not sure which signaling pathway he's talking about, but using nuclear receptors or something, I guess. My experience, even with something as simple as HIV, is that the details are still being discovered and so forth. This issue that I briefly mentioned, there were two camps about when the capsid of the HIV actually comes apart, because people looked at the size of the capsid core particle, and they looked at the size of the nuclear pore, and they said, this thing cannot fit in through the nuclear pore. And then as more and more became known about the nuclear pore complex, it went from a static model to a dynamic model. And so now people understand, or at least, and this is, still some people will debate, but the nuclear pore is more elastic than people thought. And in fact, there now are EM tomograms of intact the capsid core particles. These are these kind of oblong shaped containers intact inside the nucleus. So at least in some cases they do get through, and then they break apart. So even there, in that one of the simplest systems, there's still a lot of debate about what the mechanisms of things happen. And we're not, I see what we're doing as being more kind of tool builders in a way, trying to make it so that more people ask more questions. So I think that's important. And, once you, like I mentioned, once you put a model out and people actually understand that model is, they can understand what kind of questions to ask and what experiments to do. Yeah.
Yeah, I was wondering when you were showing the models and the interfaces to analyze them, how much information can the user get back? For example, I imagine that in some cases you're just placing molecules based on their concentration, but in other cases they are going to be close to another protein interacting because you know that they interact. Can the user know, get this kind of information in the interaction? How accurate is the backup data?
So the two parts of that question, in the sense that can this be, is the interface built to allow you to ask that question? And the answer is not yet. Although, for instance, I can, with this, what we call CellPack GPU, it has an interactive, it does have an interactive viewer, which allows you to see exactly what I showed you in that movie in real time. You can do those clipping planes in real time. You can zoom in real time. I didn't try to demonstrate that, but let's say I want to know where, given the random distribution, let's say, how often molecule A is close to molecule B. Well, I can put both of those up at the same time, and I can look at it. That you can do now, but I can't. We don't have the tool yet, but would be fairly easy to do. I've talked to Ludo about it already. To say, show me all of the pairs in which A and B are within, a 10 angstrom radius of each other, something like that. I mean, that would be relatively trivial to do, but we're still doing those kinds of developments. Yeah, but yeah, I mean, that's, the more people that use the tools, the more idea we have of what people want to do with them, in some sense, because they'll let us know. Yeah.
Sure, I'm sure you get all of the emails asking about what can be or cannot be done.
Yeah.
Yeah. Miguel?
Yeah, I was wondering if you plan, or if you are willing to plan to organize a workshop to teach people to use the systems, the modeling tools, etc. What about a workshop? Maybe we could organize one in Brazil.
Yeah, I think it's a good idea, and we haven't really done that yet. We'll see. So we have one grant that is pending right now. So David is PI on the CellPack grant, and so that was pretty much a development grant. In other words, the specific aims of the first round of funding was to develop the software. And the second round, which is not, is basically, we should know any day now whether it's being funded or not, is to do further development, like the things that Rafaela was mentioning, the kinds of probing that you want to do, and also the flexibility of the ingredients and so forth, but also support for outreach and training. But you know, I guess the one good fallout from the pandemic, if there can be such a thing, is that people are now used to having these kinds of workshops where what used to cost, you know, when
Yeah, very much.
I mean, you mentioned, Rafaela mentioned this Gordon conference I held back in, I don't know when it was, 2009 or something. And, you know, the budget for that was like $200,000. And now you can hold a workshop for almost nothing, except for the time that it takes to organize it and so forth, which is not insubstantial, but it's much more possible now.
But
yeah.
I'm sure that our coordinator Aristotle is here. He can raise the money. Right?
We will be very glad if you could possibly come presentially and stay with us at least for 1 week to give a short and concentrated course to the entire people here, the PhD and master students, the professors. I think it will be great. So I am formally inviting you. And of course, if we are inviting all the costs is by our side. So it will be fantastic. Please consider this invitation, please.
Okay, well, thank you. I definitely consider it. I mean, I think we're kind of at the stage in a number of areas. So that there's the cell pack area, and then there's the what I would call the more virtual world area, and kind of the outreach, what I would call the outreach area, communication, how you develop things for communication. All of those things, I think, would need, in a sense, separate or a more extended thought in terms of developing the materials for something like that. But it's something that we want to do. And in fact, with regard to the VR stuff that we've been developing, we're just at the stage where so up to now, I've been the only user. And last week, we put David Goodsell into it, and he was a totally naive user. He's not a gamer or anything like that. And he was impressed enough. But again, you know, you have scientists aren't typically, they're not used to using 6 degrees of freedom controllers and navigating in that world. So it was a learning experience for us in terms of how we guided him through
the virtual reality
his first experience. So we're gonna, our first step is to release it to the other members of the lab first, and let them, those that want to, play around in it, and with a guide in some sense. And then, you know, the idea of having an quote in-game guide, I think we could develop that fairly rapidly, and that might be true for the other tools as well. I'm not, I'm not that experienced in game development per se, and so forth. But I think there are a lot of tools now for making tutorials. So so we're actively looking into that, and I'm happy to talk further about it with you guys and see whether we can do something. Hopefully, even though it's so much easier in a way not to get on a plane and go someplace, it's also much better to be in person with people. I mean, especially in a learning situation, I think there's a lot that is lost through the electronic distance. But
so we are very impressive for your inspirational lecture. It was fantastic. There are lots and lots of people here in the chat congratulating you, saying that the lecture was sensational, fantastic, inspirational. So thank you very, very much.
You're more than welcome.
For this lecture, for our graduate program in bioinformatics, and for the whole PhD and master students and professors, not only from our own university, but from other universities in Brazil that are that were seeing that your lecture. Thank you very, very much.
Okay, well, my pleasure. And be in touch.
Yes, for sure.
Okay, thank you. Bye-bye.
Rafael, bye-bye.
So we have a couple questions in the chat yet. So Anderson Santos, first he congratulates you, and then he mentions that you do have a dream team of programmers, and he asks, which is the principal programming environment for these models?
Good question. So, as I mentioned, a lot of the development is being done in Unity, and Unity, and a lot of development is done in Python these days. That's not to say that we're not, so most of the, as far as the visualization stuff is done in the game engine or in the Python realm. The material I talked to today, that's really where, let's see, I think Python, I'm sorry, I think Unity uses C# instead of Python, but they're very similar kind of scripting languages, and all of them are fairly high level at this point. Now you can put in, well, I should make one caveat. Currently, David is kind of more my vintage, not quite as old as I am, but and so his lattice nucleoid model was originally written in Fortran. For those of you who are familiar with one of the classic programming languages, and I don't know whether it's been translated into C or C++ at this point. I don't think it has. But yeah, it's kind of, the standard. If the question is what languages do we use, there's a lot of Python in the lab, a lot of it in the development of the docking software, especially. But then the routines that tend to be computationally intensive are either done on the CPU in C++ or on the GPU in either CUDA or whatever the open source GPU is. I'm blanking on it right now. Yeah.
Okay, Miguel?
Yeah, Arthur, some time ago in 1995, a Japanese friend of us, Minoru Kanehisa, started a site for pathways, KEG pathways. So today, Reactome is turning the pathways into drawings and environments. So I wonder if at some point it would be interesting to have a wiki, a website that would accept, efforts of many scientists that would build models like you are doing and put them together, maybe? What do you think?
Yeah, I think it's a good idea, and I think people, it depends on kind of what the model, I mean, right now, we can export the models that we generate in CellPack and in the CellPaint VR even as SIF, in a SIF format. So that's a geometric model, and it doesn't necessarily, let's say, contain all of the metadata that you may need to modify the model per se. So, I guess the question is how much of the is, I don't know that there's a format yet. I mean, there probably is, but that contains all of the information in a compact enough form that it can be used generally. But maybe there, I'm sure people are working on that as well. But right now you can export our models essentially like you would export a PDB model. Now you don't, if I have, let's say, 1 million proteins, 1 million copies of, let's say, 1,000 different protein types, I don't want to send you a PDB file for every one of those. And so the restriction of this model would be that if there are 1,000 different proteins, I have 1,000 models, and I make instances of each one of those models, and then the scene, the model itself, from a structural point of view, is the transformation of each instance into a position in three-dimensional space. So these are rigid models. Now, the instance may itself ultimately be a flexible model, but then you have to encode the flexibility into the transformation. You may not have to, again, give every atom position, but you should be able to reconstruct every atom position. So, yeah, right now, the only thing I could say is that we export SIF files, we export Collada files, and maybe one other format that we can export. But yeah, I mean, having a repository, I think, is a good idea, but it's going to be much more complicated than,
I know.
you know, I've talked to Helen Berman at PDB, and she is averse to having models because for two reasons. One of them is they're not uniform in the data structure, and number two, they may not be based on enough experimental observation.
Well, what about PDB-E from EBI? In 2 weeks, we will have a talk about AlphaFold with the director of PDB-E, and Reactome does similar things there. So maybe in Europe they would.
Yeah, I totally agree. And you know, if something like that were to be funded or be proposed, I'd certainly
You will,
be on board with, you know, with a plan to actually implement it. I mean, these, you know, the PDB is an interesting example because it evolved over 30 years or more. From when it was first set up, it was only meant to be an archive. You know, it was a place where you could deposit your structures and be sure that those structures would be there. In other words, it was a vault to keep structures. But then obviously it's become much more than that. But it, you know, it evolved slowly. Now, models, you know, model archives are going to be much more complex.
Sure.
Yeah.
Let's think about it.
Yeah.
I think that a community that could collaborate with a wiki of information maybe would demand someone from your group programming the model, but probably it would speed up the upcoming of informations. Let's put this protein there, let's do that thing, and someone would be producing the drawing, the modeling, the, you know.
Yeah, no, I agree. And in fact, you know, David and I put together a proposal for a resource like that, but it was not funded. So, you know, it's a lot depends on, you know, factors that are beyond our control. Let's put it that way. You certainly need resources to build a resource. You need people. You know, my group is. You
know what I would suggest? A markup language, a computational code that people would contribute with a computer-friendly language. What the model has to do, you know. So if you could have a markup language, then people could contribute how the proteins would interact, and etc. And then this will feed up the model.
Yeah, no, I think it's true.
Think about a markup language, or what they say.
Yeah, well, in a way, I think Collada is kind of a markup language, and certainly CIF is a markup language. And I think the real problem is getting people to agree.
Yeah, no. We all know. But the kids, they like new things. Sometimes they will add a group to this idea.
Right.
Okay.
Yeah, it's true.
All right.
Yeah. No, this is a good discussion to have over a beer. Yeah.
Rafaela.
Well, I think we would like to emphasize thanking you for being here. Aristotle, our coordinator, had to leave at 4, but he asked us to thank you very, very much. It was a pleasure to have you here with us today. And let's hope we can plan this workshop sometime soon. It would be great if we can have you here.
You know, I don't want to jinx anything, but I know, I think the pandemic may be on the way. I don't know what it's like in Brazil, but whether your Omicron variant has swept through or not. But
we're starting to go down in the peak.
So you're probably in a similar state. In fact, Scripps is just within a couple of days changing its mask requirements if you're fully vaccinated and so forth. So things seem to be, and again, I don't want to jinx anything, but they seem to be easing up as well. So anyway, good luck. And yeah, I'd be happy to stay in touch with the group and see what we can do. Sounds good.
Okay.
Okay.
So
okay.
Thanks,
well, anyway.
Thanks a lot for this inspiring afternoon, Dr. Olson.
Oh, you're more than welcome.
It was very important for our students.
Thanks for reaching out to me. Yeah, thanks for reaching out to me. I enjoyed it.
Yeah, thank you.
Okay. Bye-bye.
Okay, bye-bye, people.
Bye.
Bye.
See you guys. Em 2 semanas, AlphaFold, gente. Até mais. Tchau, tchau. Acabou por aqui, Thiago. Encerrando?
Ok, professor.